involved in the production of proteins and amino acids. That's called as translation. So this is known as a codon. Codon is a three-lettered word which codes for a specific amino acid or a protein in a DNA. So this is known as adenine. This is uracil. This is guanine. So these three are held together in a strand. We can't read them through or single. We always read them in a multiple of three, and which sequence and along and forms proteins. This is basically the structure of DNA, which I have told you it consists of three components: sugar, phosphate, and a nitrogenous base. Sugar and nitrogenous base and sugar combine to form a nucleoside, and when these all the three are combined, they form a nucleotide. So this is the sugar, which is deoxyribose sugar in the case of DNA, and ribose sugar in the case of RNA, and this is the phosphate group, and this is thymine, and this is amine. So these bonds between phosphate and sugar are bonded by the covalent bond, and thymine and adenine and cytosine and guanine, these all are bonded by the hydrogen bond. Here thymine and adenine or adenine and thymine are bound only by double bonds, and cytosine and guanine or guanine and cytosine are bound by the three bonds in the structure of DNA. And there is a rule in codon that thymine always plays with adenine and guanine always plays with cytosine. So as we know, codon is a three-lettered word in a sequence of DNA or mRNA. There are two types of codon in DNA, which is start codon and stop codon. Start codon is always starting in DNA from A, U and G, which codes for a specific amino acids, and any three nitrogenous bases may code for any different types of amino acids or protein. There are total 63 amino acids, 63 combinations we can make from only 3 nucleotides to code very different amino acids and proteins, which are very vital for cellular functions and homeostasis. So stop codon can only be U, G and G and not any other. U, C, A, U, C, C, U, A, G. These are code for different other amino acids. But to stop the codon, while sequencing, we will sequence with each and every codon in this beginning strand. So we'll start it with AUG, but we have to end it with UGG. That's called as stop codon. And this process is called as DNA sequencing. And by that sequencing, we produce mRNA through transcription, and by that we produce proteins and amino acids with translation. Start with my topic mutations. I hope you all are clear with the DNA structure. So mutations are known as sudden changes which occur in the strands and in the codons or strands of the DNA which are responsible for changing in the amino acid production. I mean, if AUG, for example, AUG codes for phenylalanine. So if there is any change in uracil or guanine or adenine, so it may code for any other amino acid. So that would change the structure of entire protein and that would change its functionality. So the genetic code is a set of amino acids which will code for specific amino acid and not for any other amino acid. Mutations are known as the changes in the codon sequencing or DNA sequencing or mRNA sequencing. It may occur at this step or even after protein structure, 3D or 4D structure. So these are two types. Nucleotide mutations and chromosomal mutation. As I have told here about chromosomes, these are the small pairs of nucleotides which are present in the DNA strands, and this is the chromosome. So mutations can occur at any point in this. Maybe they can occur at this point or in the whole chromosome or in the semi-structure of chromosome. These are called as mutations. Now we will learn about different types of mutations under nucleotide mutation. There are 5 types of mutations in the nucleotide mutations. First we will look at silent mutation. Silent mutation is known as the change in this codon. We can change any base pair, but it doesn't involve complete change of functionality or structure of the protein. As the name suggests, that the effect of the mutation is silent in the product of the formed protein. I will give you an example. So for example, here we start reading from AUG. As I told you, this is the start codon. So we started from AUG in the translation process. So AUG will code for phenylalanine amino acid, and UUU codes for this codon codes for methionine, sorry for the mistake, and UUU will code for phenylalanine, and GWW will code for glutamic acid. So as we know that mutations are sudden changes in these codons in any sequence at any point that which will lead to change of an amino acid structure or protein structure. So if we, for example, we take cytosine, a nitrogenous base pair, and we mutate it with U. So we get AUG, UUC, and GAA. Now there is a twist here. As I have told you that 63, we can make 63 combination. So even one codon can code for other types of amino acids. So now U was there earlier. So it was coding for phenylalanine. If we change it with C, so it still codes for phenylalanine as it is the combination of genetic codes that are 68. So now this AUG is already coding for methionine and UUC is coding for phenylalanine as in the normal. Now wait a minute. This is UU. Now if you mutate, this will remain the same. UUU will become UUC because we are removing U and adding C in place of U, and GAA. Now these effects won't change because phenylalanine is coded even by UUU and UUC. So that's the reason it's called as silent mutations. As even after mutation and changing of base pairs, it doesn't change at all. That was silent mutation, and I will now go with distance mutation. So as I have told you that AUG will code for methionine and UU will code for phenylalanine and GAA code for glutamic acid. Now in distance mutations, what happens is it's just like silent mutation, but here we can see the effect of change of codon in the structure of protein. Here we have UU. So for example, if we add UU A in place of U, so here the mutated mRNA structure will be AUG, UUA and GAA. So now as AUG already codes for methionine and UUA here codes for lysine and GAA codes for glutamic acid. So now in the normal RNA in the process of translation AUG and UU and GAA was coding specifically for a X protein. Now UUU is changed into UUA. So now this will code into some other protein which will have amino acids methionine, lysine and glutamic acid, which is different than the previous one. So here, even after mutation, you can see the effects that will be resulting in changing of the structure in the final protein. These are called as distance mutations. In the DNA, GAG codes for the glutamic acid and GTG codes for valine. So for example, if we remove this A from codon and add T, so it gets mutated. So this will cause sickle cell anemia because in hemoglobin we need glutamic acid, but it's taken into valine. So it results in the structural change of hemoglobin, which leads to sickle cell anemia. Sickle cell is nothing but the RBC which are biocompetitive get changed into sickle cell, which is due to the change of hemoglobin protein. Now why did that happen? Because DNA had GAG in it and we have changed it to GTG. And even by transcription and translation, the protein which is hemoglobin got changed into another secondary structure of hemoglobin which got valine in it instead of glutamic acid. So that's the reason it results in the change of RBCs and it becomes sickle cell. Now this has both good news and a bad news because this is a disease. So good news is that malaria can't live on sickle cell and it provides immunity towards malarial parasites because they survive on the RBCs of human cells. So malaria can't live on it. But bad news is that it's a disease and we have to cure it because in the blood vessels the sickle shaped RBCs get clogged and they interfere in the normal functioning of the human body. So it has to be cured, and it's only cured genetically by changing this amino acid to GAG, so that glutamine codes for the glutamine forms the hemoglobin structure and it forms the normal biconjugate shaped RBCs. That's an example for distance mutation. So now nonsense mutation. Imagine you are all in the theater watching a movie and the movie has started and it's been 15 minutes that you are watching a movie and you suddenly see the end credits scrolling and the movie is finished just in 15 minutes. So how would your reaction be? It would be like, what nonsense is this? I came to watch a movie and it's not even, I didn't even get the time worth watching. So that would be a nonsense. That's what happens in the nonsense mutation. the sequence which is about to be like, I have told you that AUG is a start codon and it consists of a UGG, that is stop codon, which will stop coding for the RNA. But what will happen in nonsense mutation is the UGG which has to appear in the end of the translation process, it will come in the middle of any other codon and stop the further coding for the amino acid.
There is no end.
There is no end, and I'll explain you. If we add this UGG stop codon here in between of UU and GAA, so this already goes for methionine, UU goes for alanine. UGG is a stop codon here. So in a protein, for example, let's assume that a protein should contain methionine, phenylalanine, glutamic acid, and lysine. So it has to contain 4 amino acids. But here what is happening? The codon is coming in between of this stop codon is coming in between of the amino acid codons, and it's stopping the further, and it will continuously keep mutating. That's called as nonsense mutations because we don't understand really what is happening. Now we will go with the insertion mutations. Now this UG, as we know, is a start codon and it's code for vitamin. CC will code for proline and amino acid. GAA will code for glutamic acid, and UGC will code for cysteine. So now in the insertion mutation, what happens is out of all the three codons, I mean all the 3 amino acids in this codon, we only insert a single. I mean we only insert an amino acid which is not a multiple of 3. But what is the rule of genetic code? That always a codon should have a multiple of 3 amino acids. But here we are inserting only single or 2 amino acids in between of the sequence. So what will happen if it gets mutated? Let's imagine that we remove this C and we add an A here. This will be AUG, that is start codon, and if we add A in the place of C, it will become ACC. Now this A has come beside of it. So this is ACC and this C will hold it. As the rule is we have to put it on 3 codon. So this ACC and this C will be written CGA. And this AUG. And this GC, which is incomplete form. So this AUG codes for methionine and CCC codes for proline, GA codes for glutamic acid, and CCC codes for lysine. Now, in mutated RNA after translation, what will occur is AUG is however start codon, we need it for the process of making protein. Now here the CCC got mutated and A has come in the place of C. So this got moved aside. So this will change the combination of making protein. As this was synthesizing proline, this won't synthesize proline because each and every amino acid is set to a specific amino acid, to code a specific amino acid. Now this ACC will code for 3 only. Now what is occurring here? Because AUG is coding for methionine, CCC is coding for proline, GA is coding for glutamic acid, and UGC is coding for lysine. So what will occur here is AUG will however code for methionine, but here the upcoming all the codons will change the production of amino acids because here this got changed because ACC is producing threonine and this is CGA which however won't produce glutamic acid, and this is ACC which won't produce lysine, and this is GC which is an incomplete form. In this situation this GC is incomplete. So the form protein will also be incomplete and the function won't be as of normal protein.
Results in misreading. Results in misreading.
Result will be in the misreading and the protein won't form completely, which was about to be formed in the normal DNA because we have mutated a single base pair and it has changed the whole structure of the formed protein. As we know that proteins are made of amino acid sequences, this is the normal sequence. We have in protein, glutamic acid and lysine. this is a combination like A, B, C and D, but we are changing it as C, D and here. So it won't be like the primary structure which was in the DNA. This ACC codes for threonine, CGA codes for arginine. AUG codes for again threonine, as we know that this is
Initiating. Again initiating code.
no, yeah, initiating codon has got into it because of the change in sequences. This is incomplete, so all I won't code in just 2 days. So here we have been coding nucleic acid and lysine, but in this mutated gene, the protein structure is not same. So the functions, the structures will differ in both normal and mutated protein and insertion because they are inserting a single base pair which is not a multiple of 3. Now let's go to division. Let's move this C from the normal gene which goes for alanine. CCC goes for choline, GA goes for glutamic acid, and GC goes for cysteine. So now if we mutate it and we are deleting a single base pair, and again it's not a multiple of 3. What we have to do is, even if we mutate it, we have to remove the whole codon, which is consisting of 3 base pairs, and it's a multiple of 3 because there are 3 amino acids. But what we are doing is, we are just deleting a single cysteine from this cytosine. So the form mutated gene will be, after removing this C, CC and this G will pair with this codon and A here. And GC. Again, we got incomplete, just like the insertion. So now AUG will code for thymine and CCG will code for choline and AAU will code for asparagine, which was not in the case of earlier normal DNA structure. So again the protein structures will get disturbed and the function of the cell may be disrupted. These 2 mutations are called as point mutations because we are just removing one point in the nuclear base pair, but it's not changing the reading codon structure. If this is example mRNA and we are changing its C and we are just changing C, A, G and UAG. So here the reading phase doesn't shift. We are just shifted a codon, but the reading structure is same. We are not getting any other amino acids by these two. However, but even if we, let's imagine these both are same, so they both will however code for the same amino acid. So as we have made it that mutation, it won't show any effect. So these are called as point mutations because they won't have any much of an effect on a protein, and also it won't really matter if even if they get mutated in a lab in a small scale. And the nonsense mutation, insertion and deletion are called as frame shift mutations because the whole reading frame is getting shifted and the whole protein from further coding gets disrupted. I hope you all are clear with nucleotide mutations and you with these chromosomal mutations now. So here as we have seen in the structure, the changes that takes place in the nucleotide sequences of the DNA strands
Silence.
are called as nucleotide mutations, and if the same changes which are occurring in the compact and constituent chromosome structure, they are called as chromosomal mutations. And mutations can occur at any point from this DNA strand starting phosphate group until the end of the phosphate group. Deletion is known as if we have two chromatids, we have two daughter chromatids and we have to delete. Let's imagine that something happens in this chemical composition and the gene gets removed. So now the formed chromosome will look like a part of a chromosome, a whole chromosome gets removed, in which the DNA and all the base pairs will be there, it gets removed. So it will, as we know that these things contain gene in it, which are responsible for certain characteristics, called as recessive gene and dominant genes. So these genes are only getting removed from it. So it will result in very abnormal functionalities such as genetic disorders, Duchenne muscular dystrophy and all that. If we delete a part of gene, in insertion what happens? We have two different chromosomes. As we know that we have 43 chromosomes. That maybe if any two different types of chromosomes which are not supposed to link are getting linked. Let's imagine this part of the chromosome gets attached to this. So now, if this chromosome already had another pair chromatid and it had a specific X function, now these both functions will change and they will lead to a different kind of functionality in the cell which we don't desire. Inversion. What happens is in inversion, it doesn't take any other chromatids from any other chromosome. The existing chromosome itself gets in conformational structural changes, and the genes get inverted, flipped, maybe in upside down or side by side, as they have been flipped here diagonally. So that would lead to a mutation. As however we know that this changing in its structure will lead into mutation. So however, if we are changing this chromatids, we are indirectly changing the DNA structure of the chromosome. So it will lead into chromosomal mutation, that is inversion, transformation. Imagine we have to pair two of these chromatids together to form two chromosomes. And what will happen is this was supposed to join with this and form a chromosome, and the green chromosome was supposed to join with this green chromosome chromatid to form chromosome. What will happen is both the chromatids get swapped. The point at which they have to be joined at kinetochore gets swapped and they get translocated. So that's called as translocated mutation. That is all about chromosomal mutations. Now I want to end my seminar with from a single base pair change to the entire chromosomal mutation. Mutations show us how important and how delicate genetic code is to know and to cure the various diseases such as sickle cell anemia or muscular dystrophy. So some mutations may cause the complications and diseases, and some mutations may be involved in the evolution of the human life. Thank you.